everyone, and welcome to Chapter 12. This is Chapter 12 of Part 1, dealing with the nervous system. Now, we know the nervous system is a very complex collection of nerves and specialized cells known as neurons that are going to transmit signals between different parts of the body. The nervous system is fundamental for our conscious experience, personality, as well as behavior. So it's the study of the nervous system is called neurobiology, and it combines the behavioral and life sciences. Now, there are two organ systems that are dedicated to maintaining the internal coordination. That's the endocrine system and the nervous system. The nervous system deals with communications by means of chemical messengers, such as hormones, that are going to be secreted into the blood. The nervous system, they deal with the electrical and the chemical means to send messages from cell to cell. Now, the nervous system, it carries out its coordinating task in three basic steps. The first step. Now, through sense organs and sensory nerve endings, it receives information about the body and the external environment and transmits messages to the spinal cord as well as the brain. Number two, the spinal cord and the brain, they're going to process this information and then relate it to past experiences and determine an appropriate response. And number three, the spinal cord and the brain, they're going to issue commands to muscle and gland cells to carry out the response. So for the first one, you're going to have a detection. You're going to receive the information. For example, if it's cold outside, what's going to happen? Your body is going to sense it. You have various receptors that are going to sense that the external environment is cold. That information is then transmitted or sent to the spinal cord to be processed. The spinal cord and the brain are going to decide, hey, what are we going to do? This person is getting cold. Various organs are going to shut down if we don't respond correctly. So what are we going to do? When the brain makes up that decision, then it sends the information back to the muscles and the glands to carry out the response. So what are the muscles going to do? What are the blood vessels going to do? Blood vessels are going to vasoconstrict, and you'll have piloerector muscles that are going to cause the hair on your arms to stand, and of course goosebumps. So that would be the response. Now, the nervous system has two major anatomical subdivisions. They are the nervous system and the peripheral nervous system. The nervous system, what are we going to deal with? The brain and the spinal cord. Okay? And those are enclosed and protected by the cranium and the vertebral column. The peripheral nervous system now, they consist of all of the nerve system except the brain and the spinal cord. So that's everything outside here, running along the extremities, and of course our little ganglia here as well. Now, the nerve, that is considered to be a bundle of nerve fibers or axons wrapped in connective tissue. A ganglion, which is what we find here, is a knot-like swelling in the nerve where the cell bodies of neurons are concentrated. So ganglion is the singular form, and ganglia is the plural form. Now, when we look at the subdivision as a whole for the nervous system, we already said the two main divisions are the central nervous system and the peripheral nervous system. The central nervous system, the CNS, deals with what? The brain and the spinal cord. The peripheral nervous system deals with the sensory and motor divisions. Those are the two main subdivisions of the peripheral nervous system. The sensory division is divided into visceral sensory division and the somatic sensory division. The motor division, what do you think they're divided into? The visceral motor division and the somatic motor division. Now the visceral motor division is divided even further. And we have the sympathetic and the parasympathetic. You may have heard about these before. We'll get into that just a little bit later. So the peripheral nervous system, which is one we're going to deal with just for a little bit, we're dealing with their two major functional subdivisions. And already said we're dealing with the sensory and the motor. So with the sensory or afferent division, they're going to carry the sensory signals from various receptors to the CNS. So they're carrying that information from the cold weather, which will be your thermal receptors. They're going to carry that information to the CNS. That information will inform the CNS of the stimuli within and around the body. So it's going to tell you what's going on outside the body as well as inside of the body. Now, as we saw in the previous slide, the sensory division is divided into the somatic, sensory, and the motor sensory division. In the visceral sensory division. I'm sorry. So the somatic, it carries signals from the receptors to the receptors in the skin, muscles, bones, and joints. So for instance, if it's cold outside, we're dealing with the somatic sensory division. If you tore a muscle, somatic. For bones, if there's a fracture, if there's a dislocation, we're dealing with somatic sensory division. Now the visceral sensory division, it carries signals from the viscera of the thoracic and abdominal cavities. We're looking at organs that are involved in the thoracic and abdominal cavities. So what type of organs are we looking at? The heart, the lungs, the stomach, the pancreas, kidneys, gallbladder, liver. Those are the organs that you'll find within the abdominal cavity as well as the thoracic cavity. Our second major subdivision of the of the peripheral nervous system is the motor or efferent division, and this carries signals from the CNS to gland and muscle cells that carry out the body's response. So it's going to take that information that was sent to the central nervous system and send it to the gland, and the gland is either going to secrete a particular hormone, increase the secretion of a particular hormone, or decrease it. The muscle cells are either going to contract or relax. Okay? And we have effectors that respond to the commands to help the process to follow through. Now we have two divisions, as we mentioned earlier. We have the somatic motor division and the visceral motor division. So the somatic motor division, they carry out signals to the skeletal muscle. Okay, and the output produces muscular contractions as well as somatic reflexes, and they're going to involve involuntary muscle contractions, and those are called somatic reflexes. When we deal with the visceral motor division, that carries signals to the glands, the cardiac muscle, the smooth muscle over which we have no voluntary control, and those responses are called visceral reflexes. Now the autonomic nervous system, also the visceral motor division, they have two further divisions, the sympathetic and the parasympathetic. The sympathetic division, it tends to arouse the body for action. For example, through accelerating of the heartbeat and increasing respiratory airflow, and of course you're going to inhibit digestion. So if you're being chased by something or someone, what happens? Your heart rate's going to increase, your breathing's going to increase, and at the same time, while you have an increased blood flow to the various muscles that are needed to help you run, you're going to have a decrease of blood flow to the digestive and the urinary system. Why? Because you don't need it at that point in time. You don't need, your food doesn't need to be digested. You're too busy running away. Then we have the parasympathetic, which is the opposite, which is that calming effect. So this picture, you have reached to a safe hiding spot or safe space, or even back at your house, and you're in that calming effect. So your heart rate has decreased as well as your breathing. Of course, you're thirsty, a little bit hungry, so what? You're going to have an increase of blood flow to your digestive system and maybe your urinary system. But everything is in that calming state. So sympathetic deals with that fight or flight, or arousing of the body for action, and the parasympathetic deals with the calming effect. Alright, now the cells or neurons, they have three fundamental physiological properties that enable them to communicate with other cells. We have excitability, also called irritability. Number 2, conductivity. And number 3, secretion. When you do with excitability, all cells are excited. So that means that they're going to respond to a stimuli. The neurons have developed this property to the highest degree. When you do it, number 2, conductivity. Neurons respond to stimuli by producing electrical signals that are conducted to other cells. And number 3, secretion. When electrical signals reaches the end of a nerve fiber, the neuron secretes a neurotransmitter that's going to stimulate the next cell. Now, neurons fall into 3 fundamental classes based on the 3 major aspects of the nervous system function. We have the sensory or afferent neurons, we have the interneurons, and then we have the motor or efferent neurons. So let's look at the sensory neurons. Seems like the information is being repeated, but repetition is not a problem. I help you with remembering. Now with the sensory neurons, you have a transmission of information. You have stimuli that is detected by the various receptors within a particular area. It could be within the skin, within an organ, but you have receptors that are going to detect the stimuli and then send it to the CNS. Now, this information is transmitted, and it begins in almost every organ of the body, and then it ends right here in the CNS. So you see, this is the ending part. And afferent means conducting signals toward the CNS. When we look at the interneurons, these interneurons, they're going to be found inside of the CNS. And they receive signals from many other neurons and carry out integrated functions and make decisions about the response. And about 90% of the neurons in the body are interneurons. 90% of the neurons in the human body are interneurons. The word interneuron refers to the fact that they lie between and interconnect the incoming sensory pathways and the outgoing motor pathways of the CNS. Now the motor or the afferent neuron, they send signals predominantly to cells, to muscle cells and gland cells. Those are the effectors. Now they are called motor neurons because most of them lead to the muscle cells, and the efferent because they carry signals away from the CNS. So this is your A. This is your S, your sensory, also called the afferent. This is your I, also called association neurons, and this is your M, So to help you remember the three fundamental classes, you can use SIM, S-I-M, sensory, interneurons, and motor, and that is the order. You're not going to have motor, then sensory, then interneurons. No, that's completely incorrect. Remember that order. Now let's take a look at the neuron itself. We have the soma. What is the soma? Well, the soma is considered the control center. It's also called the neurosoma, the cell or the cell body. Now it contains a single centrally located nucleus, which you see here. Here's our soma, and it contains a nucleus with a very large nucleolus, which you see right there inside of it. Now the cytoplasm contains mitochondria, lysosomes, Golgi apparatus or Golgi complex, and many inclusions, and an extensive rough ER or rough endoplasmic reticulum and cytoskeleton. Now the cytoskeleton consists of a dense mesh of microtubules and neurofibrils that are compartmentalized. The rough ER into dark staining nasal bodies. See that move there. Alright, so mature neurons, they do not divide, but they have long lives. Now stem cells in the CNS, however, they can divide and develop into new neurons. The major cytoplasmic inclusions are glycogen granules, we have lipid droplets, melanin, and a golden brown pigment called lipofuscin. Now lipofuscin is produced when those lysosomes start to digest worn out organelles. So when these organelles are a little bit aged, have a little bit of age on them, the lysosomes, the lipofusions, they are produced when the lysosomes start to digest these old looking organelles. Now the lipofusions also accumulate with age, and pushes the nucleus to one side of the cells. So the granules of the lipofusions are called wear and tear granules. Now the soma gives rise to these very thick branches here, and they're called dendrites. They look like tree branches that are like extending off the root, or they may look like the root of a tree, but these little extensions off the soma are called dendrites. Now there are a number of them, and they vary from thousands depending on the neuron, and they're usually as tangled as multiple dendrites may seem. They provide highly precise pathways, and they are those pathways for reception as well as processing of neural information. Then we have our axon running along here. Now the axon, it originates on the mound on a soma called the axon helix. So here's our axon helix, and our axon originates from there. So it says that long extension going all the way down. Now the axon is cylindrical and relatively unbranched for most of its length, although it may give rise to axon collateral, which you see this branch here, along the way. So along the way, depending on the type of okay, so let me continue to look at the axons. Most axons, they branch off extensively at their distal end. So at this particular end, you're going to see them branching off here. Okay. Now an axon is specialized for rapid conduction of nerve signals to points remote from the from the soma. Let's turn this on. Okay, so it's cytoplasm of the axon is the axon plasma, and its membrane is the axolemma. Now, a neuron never has more than one axon, and some have none. Now, it's Schwann cells. Now, there are Schwann cells and have myelin sheaths that are associated with the axon. Okay. Now, somas, they're going to range from 5 to 130 micrometers in diameter, while the axons, they're going to range from approximately 120 micrometers, and from a few millimeters to more than a meter in length. Now, at the distal end, down here, of an axon, it usually has a terminal aberration of fine branches. Now, each branch ends in a synaptic knob. Okay, so those are these little knobs that you see at the end of the branch. And then that forms a junction or synapse with the next cell. Now, the synapse contains synaptic vesicles full of neurotransmitters. Now, in autonomic neurons, the axons has numerous beads called varsicosities. Now, the neuron structure varies, and they are classified according to the number of processes extending from the stroma. We have a multipolar neuron, and with that multipolar neuron, those neurons are those with one axon. You see here, one axon and multiple dendrites, and they are the most common type of neuron. The bipolar neuron, they have one axon and one dendrite. Now, those ones you might find in the inner ear, as well as in the retina or the olfactory cells. Then we have the unipolar, and they only have a single process leading away from the soma, and they are represented by the nucleus that carry signals or sensory signals to the spinal cord. Now, since it's only a short distance away from the soma, the process branches, they look like a T, and with a perpendicular fiber bringing signals from a source of sensory and a central fiber continuing into the spinal cord. Now, the axonic neuron, they have multiple dendrites but no axon. So they're going to communicate through their dendrites and do not produce action potentials. And some are found in the brain, the retina, as well as the adrenal medulla. Alright, so many proteins are made in the soma. They must be transported to the axon and the axon terminal. And they're there to repair the axolemma, and they serve as gated ion channel proteins, as enzymes, or as neurotransmitters. The axonal transport refers to the passage of proteins, organelles, and other materials along an axon to and from the cell soma. Now, movement away from the soma down the axon is called anterograde transport. So that's coming from from the soma down the axon. That's called our anterograde transport. And then our retrograde transport is movement up the axon toward the soma. Now, materials, they travel along microtubules of the cytoskeleton. Now, we do have a little motor protein for the retrograde transport, and it's called the kinesin. Now, the motor protein for the retrograde is called the dynin. And they act somewhat like a myosin head of muscle, of the heads of the muscle, and they crawl along these microtubules. Now, there are two types of axonal transport, and they are fast and slow. The fast axonal transport occurs at a rate of 20 to 400 millimeters per day, and may be anterograde or retrograde. For the fast anterograde, it transports mitochondria, synaptic vessels, other organelles, axilla components, calcium ions, enzymes, and they carry those things towards the distal end of the axon. So that's all the way down here to the end, from the soma all the way down to the end. The fast retrograde, so that's returning from the axon all the way to the soma, their transport returns. It uses the synaptic vesicles to the soma, and informs the soma of any conditions at the terminals. So if there's any type of malfunctioning going on here at the axon terminals, that information is sent back to the soma. Now, some pathogens, such as tetanus toxin or certain viruses, they invade the nervous system by entering the distal tips of the axons, and begin transported to the soma by the fast retrograde transport. Now we have the slow axonal transport, also called the exoplasmic flow, and that occurs at a rate of 0.5 to 10 millimeters per day, and is always anterograde. Now, the slow axonal transport, it moves enzymes and cytoskeleton components down the axons. It renews worn out axoplasmic components in the mature neurons, and then it also supplies these new axoplasm for development or the regenerating of the neurons. Now, if there are damaged nerve fibers, they are regenerated at a speed governed by the slow axonal transport. Now, neurons, they're outnumbered by as much as 50 to 100 by supported cells called neuraglia, or glia cells. Now, glia means glue, and one of the roles of these cells is to bind neurons and form a supportive framework. Let's move on. Alright, so we're going to take a look at the different types of cells. We have astrocyte, we have oligodendrite, then we have our ependymal cells, and we have our microglia. Alright, so look at the four types of neuroglia that we have. The first one is oligodendrocytes. Now, they look like an octopus. We saw which one they look like right here in the middle here. Okay, so it looks like an octopus with as many as 15 arm-like processes. Now, each process, they're going to spindle around the nerve fiber, just like an electrical tape. It's going to wrap around the fiber with insulating myelin sheath. So it's going to have a myelin sheath within to help insulate it. Now this sheath, it speeds up the nerve conduction. Then we have the ependymal cells. They resemble cuboidal. Here we have them right here down here. They look at cuboidal epithelial lining the internal cavities of the brain as well as the spinal cord. So remember, I hope you guys remember from last semester, from 201, where we talked about cuboidal and columnar, talking about the shape of the cell. So this one is cuboidal epithelium. Now, however, they have no basement membrane and have no root-like processes that are going to penetrate the underlying tissues. Now, these cells, they produce CSF, which is a cerebrospinal fluid, which are going to bathe and lubricate the CNS and fill into the internal cavities. Now, these cells have cilia, and they help to circulate this CSF. The microglia, which is our third one, these are small macrophages that develop from white blood cells called monocytes. So here is our microglia. Now they wander through the CNS, constantly probing for cellular debris or other problems. So these are like the generals or the guards of the CNS to make sure nothing is getting defective. Those are our microglia. They are thought to perform a complete backup on the brain tissue several times a day, phagocytizing dead cells. That means engulfing them and getting rid of them. Dead tissue as well as microorganisms, things that don't belong there. They're going to engulf it and get rid of them. Okay, they become concentrated in areas damaged by infection, trauma, or stroke, and pathologists look for clusters in the brain tissue as a clue for any type of injury. So if you see them clustered together, there is definitely something occurring within that particular area of the brain. Okay. Then our last one is the astrocytes. Astrocytes are the most abundant glia, and they constitute over 90% of the tissue in some of the brain area. They are named for their many branched star-like shapes. They have most diverse functions of the glia. They form a supportive framework for the nervous system. They have extensions called perivascular feet, okay, that contact the blood vessels, and then they stimulate them to form a tight seal called the blood-brain barrier. So here are our astrocytes, and you see the little perivascular feet that are attached to the blood vessels. Okay. They also monitor neural activity, and they signal blood vessels to constrict or dilate to regulate blood flow, provided oxygen and nutrient supply in accordance to the neural demand. They convert blood glucose to lactate and supply it to the neurons. They also secrete nerve growth factors, proteins that promote neuron growth and synapse formation. They communicate electrically with neurons and may influence signaling. They also regulate the chemical composition of the tissue fluid, absorbing neurotransmitters and potassium ions so they do not reach excessive levels. And they form hardened scar tissue when neurons are damaged. And that process is called astrocytosis or sclerosis. Now we have these things called Schwann cells. And there are only two types that are found within the PNS, the peripheral nervous systems. Now the Schwann cells is the Schwann cells, and the satellite cells. The Schwann cells, they're the ones that they're going to envelope the nerve fibers of the PNS, and they wind repeatedly around a nerve fiber, and they're going to produce the myelin sheath similar to the one produced by the oligodendrocytes in the CNS. Now the Schwann cells, they also assist in the regeneration of any damaged fibers. The satellite cells now, they're going to surround the neurosoma or the soma in the ganglia of the PNS, and they provide electrical insulation around the soma, and they regulate the chemical environment. So we have the satellite cells that are surrounding the soma, while the Schwann cells surround the nerve fibers, and these two are the only ones that you'll find in the PNS. Now when we look at tumors, tumors are masses of rapidly growing or rapidly dividing cells. Mature neurons that have little or no capacity for mitosis and seldom form any type of tumors. Now tumors, they arise from meninges, which are that protective layer of the CNS, metastasis from neuronal tumors in the other organs, as well as they often ganglion cells that are mitotically active throughout life. Now gliomas, they grow rapidly and are highly malignant, and these blood-brain barriers, they decrease the effectiveness of chemotherapy. And of course, treatment consists of radiation or surgery. Now when we look at the myelin sheath, it's an insulating layer around the nerve fiber, somewhat like a rubber insulating a wire. Now because the myelin sheath, or the myelin, is a plasma membrane of the glial cells, its composition is like that of the membrane, with about 20% protein and 20% mixed lipids. Now the production of the myelin sheath is called myelination. Then it begins in the fourth week of fetal development, in the 14th week of fetal development, and it proceeds rapidly in infancy and isn't completed until late adolescence. Now children under the age of 2 may not be put on a low-fat diet because this could interfere with the myelination. Now in the PNS, the Schwann cells, the Schwann cell spirals rapidly around a single nerve fiber, and they're going to lay down as many as 100 compact layers of its membrane with only no cytoplasm. The Schwann cells, they spirals outward, finally ending with a thick outmost coil called the neurolema, and that which consists of a body of Schwann cell with its nucleus and most of its cytoplasm. Now the external to the neurolema is a basal lamina, and then a thin sleeve of fibrous connective tissue called the endoneurium. So let's take a look at it right here. Here we have the nerve fiber, and of course, covering the nerve fiber, we have the axonal. Then we have the neurolema. What do we say the neurolema is? It's that thick outermost coil. Okay. and it contains these Schwann cells with a nucleus. And this consists of your myelin sheath. So here's that little process I was referring to as to what happens when you have this myelin sheath. I mean, not the myelin sheath, sorry, the Schwann cells. So here's your axon. Again, here's another diagram of what we're talking about. Here's your axon. And around the axon, you have the Schwann cell that's going to spiral around it. When it finally spirals, you know you have the nucleus that is embedded within this Schwann cells. As it keeps spiraling around, we have that external portion to the neurolemma, which is the basal lemma. Here's that basal lemma here in that light purplish area. Okay? And then on top of that, we have a very thin sheath of fibrous connective tissue called the endoneurium. Okay? Now in the CNS, each oligodendrites, they're going to reach out to myelinate several nerve fibers in its immediate vicinity. Because it is anchored in multiple nerve fibers, it cannot migrate around them. So it pushes newer layers of myelin under the old ones. Myelination spirals inward toward the nerve fiber. The nerve fibers of the CNS has no neurolemma or no endoneurium. So that outer sheath that was covering, it's not there. That was covering the basal lemma, lamina or the basal lamina is not going to be there, that outer sheath. Okay? Now many Schwann cells or the oligodendrites are needed to cover one nerve fiber. Now each sheath is segmented into different parts. We have the nodes of Ranvier, we have the internodes, and then we have the initial segment, and we have the trigger zone. Now the nodes of Ranvier, those are the gaps that you're going to find right in here. Those gaps that you're going to find between the segments. The interneurons, the internodes, that's not myelin covered segments from one gap to the next. The initial segment, let me go back, the initial segment, that's that short segment of nerve fiber between the axon helix and the first ganglia. So that small little segment section right here, that's the initial segment, because this section here is the axon, the axon helix, and then our trigger zone, that is the axon helix plus the segment section. So from here, from the tip of the very first ganglia to the beginning of the axon helix, this is considered to be the trigger zone. So that's the axon helix plus the initial segment gives us our trigger zone. Okay, let's skip over to the next page. So let's talk about the diseases of the myelin sheath. Now, we have a disorder called multiple sclerosis. Everybody has heard of that, and it deals with when the oligodendrocytes and the myelin sheath are deteriorating, and those are the ones that are deteriorating in the CNS. The myelin is then replaced by hardened scar tissue. The nerve conduction is disrupted, and that's where you have double vision, the person starts to tremor, and the speech is also defective. Now, most people get an onset between the ages of 20 to 40, and usually are fatal from ages 25 to 30 years after being diagnosed. If you're diagnosed with it now, and then it may become a little bit fatal 20 to 25 to 30 years from now. Of course, it may be caused by an autoimmune triggered by a virus. Then we have TaySachs disease, which is a hereditary disorder in infants of Eastern Europe, Jewish ancestry. And it's an abnormal accumulation of glycolipid called the GM2 in the myelin sheath. And it causes blindness, loss of coordination, and as well as dementia. Now, when it is accumulated, when that GM is accumulated, it causes a disruption in the nerve cells or the nerves. Alright, and if a person obtains this particular disease before the age of 4, it can be extremely fatal for that particular individual. Alright, so let's look at these unmyelinated nerve fibers. Now, many nerve fibers in the CNS and the PNS are unmyelinated. But in the PNS, even unmyelinated fibers are enveloped by the Schwann cells. We have them right here. Sorry, not the outer part, the ones on the inside. Now, one Schwann cell is going to harbor at least 1 to 12 small unmyelinated fibers. So here we have these unmyelinated fibers right in here that are surrounded by the Schwann cells. Now the Schwann cells, their plasma membrane, it doesn't spiral around the fiber, but it folds once and somewhat overlaps the edges of the fibers. Okay. Now this wrapping is the neurolema, also called the mass axon in the unmyelinated fibers. Now, most nerve fibers have individual channels, but small fibers are sometimes bundled. Now, a basal lemma, which is what we have, let me see if I can get this going here. The basal lamina, which we have on the outside, it surrounds the Schwann cell and the fibers. So here are the Schwann cell, and here are our basal lamina, and there are unmyelinated nerve fibers. Now, the speed at which nerve signals are conducted along a nerve fiber depends on the diameter and the presence of the absence of myelin. Now, the signal conduction occurs along the surface of a fiber, not in the axon plasma. So it's on the outside, not on the inside, is where we have the signal conductions. Now, large fibers, they have more surface area, and conductions signals are more rapidly than the small ones. And myelin further speeds signal conduction. Now, when we look at the nerve signals, they travel about 0.5 to 2.0 meters per second in small unmyelinated fibers, and 3 to 50 meters per second in myelinated fibers of the same size. Now, in large myelinated fibers, nerve signals, they're going to travel as fast as 120 meters per second. And we have these slow unmyelinated fibers. Those are sufficient for many purposes, such as pupil dilation and supplying the stomach. Now, the fast myelinated fibers, they occur where speed is vital, such as in motor commands to the skeletal muscle. Those are good for signals for balance and vision. Now, nerve fibers of the PNS are very vulnerable to trauma. So remember, we're talking about the PNS. These are talking about these nerve fibers on the extremities, so the fingers, the legs. So they're more vulnerable to any type of trauma, and but may regenerate of its soma may regenerate if its soma is intact and at least some neurolemma remains. So I'm going to move all the way down to go to a slide. This is just talking about the process of how the nerve fibers regenerate. So I'm going to go to this particular diagram here. Okay, this diagram is showing you the very first one. Talks about a normal nerve fiber, exactly what it looks like. It has an intact endoneurium, myelin sheath, and here we have the muscle fiber, and we have the neuromuscular conjunction, or not conjunction, I'm sorry, the neuromuscular junction. So what's going to happen when there is some type of trauma? We're going to go through the various steps. Now, pretend that the fiber is severed, or the nerve fiber is cut. Now, what happens at the distal end to the injury? It cannot survive because it cannot synapse. So this part cannot synapse. This is the distal end of the fiber. It cannot synapse properly. So the terminal is going to start to degenerate because information is not being transmitted to the muscle. So we're going to have a protein synthesis. Organelles are limited to the soma. We have the Schwann cells of the distal fiber also start to degenerate, which I just mentioned. You see this, get this mouse moving. Also starts to degenerate. And we have macrophages that clean up tissue debris at the point of the injury. So we're going to have these little macrophages that are going to come out and start cleaning up the mess. So we call these the maids. These are the macrophages. Okay. Now the third one, what happens here? You notice there's a change in the soma itself. So the soma is going to start to increase in size. It's going to swell, and the endoplasmic reticulum, it starts to break up. So the nasal bodies, they start to disperse. So it's going through some type of abnormality, the soma. The nucleus is not centered anymore, so it's going to move off to the side, and some neurons, they start to die at this particular stage. Now the axon stump, it may sprout multiple growths, which we see here. We have multiple growths processing while the distal end starts to, or continues to, degenerate. Okay, so what else is happening at this particular stage? The muscle fibers, they're going to start to be deprived of any type of nerve supply, and then start to shrink. So you see that the muscle fibers are shrinking because they're not getting any type of electrical stimulation to them. And this is called, that process is called denervation atrophy. Denervation atrophy. Okay? So here we are now on step 4. Now near the injury now, we're going to have these Schwann cells, the basal lemma and the neurolemma. They start to form a particular tube, a regeneration tube. The Schwann cells, they're going to produce cell adhesive molecules and nerve growth factors. They're going to enable the nerve to regrow. Now when one axon growth process finds its way into the tube, it grows very, very rapidly, about 3 to 5 millimeters per day. That's pretty fast. Now the regeneration of this tube is going to guide the growing sprout back to the original. You see how the tube is guiding these sprouts that had start to grow out. We saw here in step 3, it's going to guide it back to the original target cells, re-establishing the synaptic contact. So we're going right back into the axon, so they can get signals to the muscle. So here it is in this particular phase here. We're on step 5. I'm sorry. Step 5, here we have it grown back in, and it's starting to get or grow or continue its growth process to the muscle itself. Now when contact is established, so when these particular these sprouts have been rerouted and is going towards and have made contact with the muscle cells, we notice what happened over here. The soma shrank, went back to its original size and appearance, and we have the re-innervation of muscle growth. So the regeneration of the muscle fiber. Here we have the denervation atrophy. This is a muscle due to loss of nerve contact by a damaged nerve. So this is what happens to the muscle cells, the muscle tissue, when there's damage to the muscle. When there's damage to the cell itself. So the whole process we're talking about, when you saw in step 3, 4, and 5, where we have atrophy of the muscle, this is what it would actually look like. Okay. All right. So we have nerve growth factor. When we look at nerve growth factor, it's a protein secreted by a gland, muscle, and glial cells, and is picked up by the axon terminals of the axon. It prevents the programmed cell death in growing neurons, and it also enables growing neurons to make contact with their target cells. And it was illustrated by Rita Levi Montelcini, back in the 1950s, and she won a Nobel Peace Prize with Stanley Collin in 1986. And they use growth factors. The use of growth factors is now a vibrant field of research. We have some other individuals. We have Galen. We have Renee. We have Luigi and Camillo. These names are very difficult to pronounce, so please forgive me. Now, the very first individual, Galen, he thought that the brain pumped a vapor called psychic pneuma through hollow nerves and squirted into the muscles to make them contract. Now, that's very interesting, but at least he had an idea of how something, at least how the brain would have worked. It was incorrect, but it was an idea. Then we have Renee in the 17th century supported this individual's theory. Then Galvani, he discovered the role of electricity in muscle contraction in the 18th century. And then by the 19th century we had Camillo Golgi, who developed an important method for staining neurons with silver. Our last individual here is Santiago Roman E. Cajal, set forth the neuron doctrine of nerve pathways is not a continuous wire or tube, but a series of cells separated by gaps called synapse. Okay. Now, there are two key issues in neurophysiology, and they are: how does a neuron generate an electrical signal, and how does it transmit a meaningful message to the next cell? So, So this is the end of our chapter, chapter 12, part 1. Please go over the video. If you have any questions, please do not hesitate to post on Blackboard. the question and answer area and put chapter 12 part 1 if you have any questions pertaining to this section. Okay, now when you have time this week, please go ahead and watch chapter 12 part 2. Okay.